This talk is on digital holography for personalized cancer therapy selection. First, I'll describe how disease is not just a change in state, but it's also a change in motion, and we can pick up those motions using intracellular Doppler spectroscopy through holographic optical coherence tomography, or OCT. Then I'll describe clinical trials that we're performing for cancer therapy selection. The idea that disease is a dynamical system really came from complex dynamics theory, and what we're really going after is altered health and therefore altered motions. Now, in complex systems theory, one thing you can do is look at networks, and this is an example of the human disease network, which shows a comparison of the states among lots of different diseases, and these are sort of static. But you can also look at another type of network. This is a signaling pathway where there's some kind of a signaling event at a receptor on the cell surface, and then there's a cascade of all these downstream proteins. And you don't really need to zoom in on these downstream proteins. What you can notice is that a lot of them relate to motion. For instance, cytoskeleton reorganization, membrane traffic, cytokinesis, membrane ruffling. So all of these motions are things that we can pick up using Doppler spectroscopy. So I want to set the stage first. So this is the graph of what we're working with, and so this is velocity as a function of size. So the smallest things, like organelles, move the fastest, and they move in the range of microns per second. At the largest scale, we come to the size of the cell. Now this is really rheology. This is shape changes in the cell as it responds to its force environment, and those speeds are often in the range of nanometers per second. Now you notice that on this log-log graph, there's a rough scaling relationship, and what that is a generalized Stokes law of active transport. And so the velocity is responding to a force. Forces in the intracellular regime are in the range of piconewtons. Now there's a viscosity which is size dependent, and so that this is actually not a inverse law, but it actually has a different power. But the bottom line is that the velocities are in the range from microns per second down to nanometers per second. And that's all because of this crowded cytosol. I also want to point out that everything that we're dealing with in this talk is active transport. So this is actively driven motion, driven by energetic molecules like ATP or GTP. And so while they are random walks, this is not thermal Brownian. And so we are not dealing with thermal Brownian motion, but we are going to be looking at directed transport. And it's the directed transport that we can pick up with Doppler light scatter. For the Doppler effect, there's a k-vector of the photon coming in, and then it scatters off, and so you have the initial and the final k-vectors scattering off of this object, which is moving with the velocity v. If you look at the change in the k-vector, that is called the momentum transfer, q, and q is largest for the backscatter orientation when phi is equal to 180 degrees. The Doppler frequency shift is given by the dot product between the q, the momentum transfer, and the velocity. And again, this is going to be largest in a collinear configuration when theta is 0. And so it's either going to be with the particle moving away from the light source or towards the light source. To put some numbers on this, if lambda is 840 nanometers, and I have a backscatter configuration, and the motion is collinear with the q vector, then for a velocity of 1 micron per second, that produces a Doppler frequency shift of about 3 hertz. Now, of course, anything that is moving inside the cytosol can be moving away or towards the light source, and so obviously the average Doppler effect is going to be 0, but you would still get these Doppler peaks, and so that this would be for the velocity moving away from in this case towards the source, and then the velocity moving away from the source. But in fact, it's not just the fact that the average Doppler frequency is 0. You also have to average over all angles. And when you do that, the Doppler peak turns into a Doppler edge. You'll notice that there's even a DC spike here. And so what's happening is that there's a lot more ways you can be transverse to the q vector than collinear with the q vector. Now this Doppler edge is something that we would then see in the sideband. And so we would look at just positive frequencies, and we'd notice that we had a plateau and then a knee frequency and then a fall off. I do want to point out that for the Doppler edge, the slope on this is much steeper than what you would see if this were Brownian motion. Now, in fact, in the cell, there's all these different processes going on. And so there's all these different speeds moving in all these different directions. And so this is just a schematic of showing roughly what the spectral power density looks like in light scattering from living tissue. And so these are all a lot of Doppler edges. And so these are falling off faster than diffusion. But these all get added together, and what you get is this very diffuse spectrum. And so it has a plateau, it has a Nyquist floor. This actually depends on your detection bandwidth, which is set by your exposure time. There's the slope. There's also, most importantly, this knee frequency. That is a good measure of the average speeds going on inside of living tissue. And so we're going to be looking at this curve and seeing what effect drugs have on that. All of this has been written up and is explained in great detail in a recent review article that I published just this year in 2024 in Reports on Progress in Physics. And so it's coherent light scattering from cellular dynamics in living tissue. This gives all types of different ways that different types of motion can scatter light, and then how you would be able to interpret that. All right, so we're also doing optical coherence tomography, or OCT. OCT is a form of laser ranging, and one of the easiest ways of describing that is with in the time domain with pulses. And so this is an example where a pulse laser puts out a laser pulse. It goes into a turbid medium, but let's say that there are these structures which scatter, and so that'll actually give what's called a coherent return. That's these little blips. It's like radar. These are like little echoes coming from this pulse that went in. But because of all the diffuse scattering going on, there's also this incoherent background that these coherent returns are sitting on top of. And this is not drawn to scale. The background could be a million times brighter than these little coherent returns that we're getting from these structures here. To expose those or extract those, what one does is you take a pulse from the same laser and you sweep it across this distributed time of flight. And every time that this overlaps, you're going to get little interference fringes, and they're going to be very dim, but you can pick them up with off-axis digital holography, and so you can select photons that are coming from different depths inside of the scattering medium. This is an example of a photon Monte Carlo, and so if I have just a line of photons coming in, they do scatter and they produce this rough cone that you see here, although you see that most of the density is actually going in, and so there's a lot of forward scattering. And then let's say it hits a dynamic target, and then it backscatters. Well, it's going to pick up a Doppler frequency, and then it carries that Doppler frequency with it as it forward scatters back out through the tissue. And so this is Doppler tagging of deep light. And by deep, I mean that we're going as deep as a millimeter, although typically we work about a half millimeter inside a living tissue. And so this is a way that we get the Doppler effect coming out, and all the different processes that are going on all at the same time. All those Doppler effects are beating against each other, and it actually creates noise. But we can do fluctuation spectroscopy, which is really just looking at the sideband spectrum that I showed in an earlier slide. So this is the setup, the optical setup. We work with a low coherence light source. This is a superluminescent diode. It's at 840 nanometers. This goes and hits a target. This will be some kind of living tissue up to about a millimeter thick, and it'll backscatter into this signal arm. There's a Fourier plane here, an image plane, but we put the camera on a Fourier plane. And then the reference is coming through this delay line, and then it's coming in off-axis, so this is off-axis holography on the Fourier plane. And then by changing these mirrors here, just by moving these back and forth, we can select light coming from different depths inside of the target. So here's an example of the full hologram. This is a blow-up. You can see the stripes on the speckle. We just do a simple FFT because we're on the Fourier plane. That produces the sideband. You zoom in on the sideband, and that is an optical section. It's about 20 microns thick because of our coherence length of our light source, and also because of multiple scattering. But it's a 20 micron thick optical section, in this case of a tumor spheroid. And so you're able to pick up the motion, and so this is actually a movie now. And so what we do is we sit at a given depth with these mirrors fixed at a certain location, and then we just take 2,000 frames. And so this is looping, but what you're seeing is the raw data on the camera. And so you have the speckle has these stripes. The stripes are the off-axis hologram. But all of this noise that you see here is all the Doppler frequencies beating against each other. And so if you just look at a single pixel in this, what you see is a time series. And so this is a fluctuating time series. So one of the simplest imaging modalities we have, we call motility contrast imaging, or MCI. And all we're doing here is we're just color coding on speckle contrast, which is just the normalized standard deviation of those fluctuations. And because we are doing laser ranging, it means that we can look at these sections as we push through, in this case, a quite large tumor spheroid. This is about a millimeter in diameter tumor spheroid. And it's so big that it has a hypoxic and necrotic core. And so here's the, we're actually coming up from the bottom. This is the bottom of the tumor. We're pushing upwards. This is roughly at the midsection. And again, the red is color-coded on just fluctuations or Doppler beats. And you can see you have the proliferating shell surrounding this hypoxic and necrotic core. So we're using motion, living motion, as our image contrast. And we can stack these up, we can do cutaways, and we can also apply drugs. For instance, here's an example sitting on a midsection of a tumor spheroid. This is the initial case. Then we apply the drug nocortisol, which is a microtubule stabilizer, and so it shuts down mitosis. And you'll notice that the activity in that proliferating shell actually decreases over time surrounding this hypoxic core. And so we can generate dose response curves this way. Well, in that case, we had fairly low resolution, but we also can work at higher resolution. This is a biodynamic microscope. It fits in an expansion slot of the Olympus X71 microscope. And so here's an example of higher resolution data. And so our voxel now is about 15 microns on a side. This is motility contrast data, so you'll see reds and blues. You see the blues in the core there, and then the blues disappear as you come up to the shell, and then we're going to fly back. And again, notice those blues. Okay, so that's the hypoxic core, but it's not as uniformly blue as it was when we had the lower resolution. So now we're actually picking up rafts of cells that are alive inside of this hypoxic and necrotic core in this tumor spheroid. All right, so if you have a fluctuating time series, you can do a lot more than just normalized standard deviation or speckle contrast. You can also do a spectral decomposition and looking at the power spectrum. And this is really our choice. But through the Wiener-Kindchen theorem, you also can do an autocorrelation trace. We like the power spectrum because there's a one-to-one relationship between speed and frequency. And so you can just read off, knee frequencies and then very quickly get an idea of what kind of speeds are involved. And also, as drugs shift this knee, if it shifts higher, it means that things are moving faster. If it shifts lower, things are moving slower. So it's very easy to interpret, and it also matches with the analysis that I described earlier in this talk. So this is how we look at a drug response. And for instance, the red curve here, that's the initial power spectrum before we apply a drug. Then we apply a drug, and then each of these gray curves is just some time later. And typically it'll be either 20 minutes later or 40 minutes later. And then the blue curve is the final spectrum that we took. So here's the final spectrum. And what we can do is we can stack these now into what we call a differential spectrogram. And so what this is we take a difference of logs. We take the log of one of these curves and then subtract off the log of the spectrum at t equals 0, which is the baseline. And then we just stack those up in this y direction here. And so this is a differential spectrogram that captures the drug effect on these spectra. In this case, the frequency is going from 10 millihertz up to above 10 hertz. And there are two sort of time axes. I mean, there's the time axis as we're working at 25 frames per second, taking a stack of frames. But then there's also this slow time axis, which is every 20 minutes we're taking a new spectrum. And so this is the slow axis here. And then this is the Fourier transform of the fast time axis. You'll notice that up to the application of the drug, this is all green in here. Well, that's because that's the baseline. And so you're getting 0, okay? And then the green is the 0. And this is fractional change. So 0.5 really does mean a 50% change in the spectral density of the spectra. This is a pretty potent drug, valinomycin at 50 micromolar, which is a pretty high dose. And so there's an immediate change that occurs at the low frequencies, but there's a slower change that occurs in the higher frequency. And then there's this mid-frequency suppression. So these are enhancements at the low frequency and the high frequency. Well, we can do more than just look at an average spectrogram. What we can do is spatially resolve it. So we can go inside and look at these spectra or spectrograms coming from different regions. In this case, this is an esophageal, it's a human esophageal biopsy. it's a cancer biopsy. And so if we look in this region here, we get what's called a red shift because you see you get enhanced low frequency, so that's the red shift. But in this region, it's a mid-frequency enhancement. And so what we can do is create biomarkers like red shifts and mid-frequency enhancements, and then we can create color merges. Now these can be positive or negative, and so we do a 4-color merge on 2 biomarkers, and it creates these very pretty plots down here. Now these are all human esophageal cancer pinch biopsies. They're now color-coded on different biomarkers, and what you notice is the heterogeneity. And so there's just a tremendous amount of heterogeneity within cancer. And these are the drug responses, right? So these biomarkers relate to how the tissue responds to a drug. And so different parts of this tissue, these different biopsies, are responding differently in different regions to the drug. And so that cancer heterogeneity is one of the biggest challenges in finding an effective therapy for cancer patients. Now we can average over the whole tissue. There is usually a good average spectrogram. So even though there's the local heterogeneity, it does produce fairly good averages over a single biopsy or a tissue sample. In this case, many of these are cancer B-cell lymphoma biopsies. Those are biopsies of lymph nodes. And a lot of these drugs are actually what are known as salvage drugs. So this is after the dogs have failed their first-line chemotherapy for B-cell lymphoma. The veterinarian is now trying to find a salvage drug that'll help the dog. And so the doctor doesn't know. It's really a coin toss. And these are the different drugs that they can apply, like actinomycin, mitoxantrone, cisplatin, carboplatin, cytarabine. And notice how their signatures, these spectrograms or fingerprints, are different for all these different drugs. There are actually a couple of cell lines here. These are DLD1 cell lines, and this is nocodazole and Taxol. But still, we can build up these libraries on the average effect of the drug on tissue. But what we really want to try to do is dig deeper into mechanism of action. Like, for instance, I'm going to talk just about cytochalasin. So cytoskeletal drugs are good reference compounds or tool compounds because they have very specific effects on the mechanics of the cell because they affect the cytoskeleton. So let me just zoom in on cytochalasin. Well, cytochalasin destabilizes actin, and therefore one of the things that cytochalasin does is it actually degrades the actin cortex, which gives the cells their stiffness. And so when the cells lose that stiffness, they become floppier, and so you'll have enhanced membrane undulations, and that's actually showing up in the mid-frequency. And so using this tool compound is one reason that we know that active membrane undulation effects are actually in our mid-frequency range. Whereas blubbing, for instance, we know from other studies that blubbing is actually in the low-frequency range. And so that way you can use these tool compounds to actually get at mechanism of action for these drugs. This is another example. This is apoptosis. What helped us here is we had fairly large tumor spheroids, and so we could look at the spectrum or spectrogram in the proliferating shell and then compare that to the spectrogram in the hypoxic core. I do want to say that I've flipped my axes here. So the fast frequency axis is here, and then the slow time over hours is along the horizontal now. But this is the high frequency. So this is the organelle transport band that we pick up in the proliferating shell, but it's missing in the hypoxic core. I mean, there's a little bit there. But in fact, the hypoxic core doesn't have the energy to go into apoptosis. So it actually just goes into necrosis. And so you get just blubbing of the cells. And so that's this showing up here in this low frequency. Whereas under apoptosis, you have active organelle transport as the cell disassembles itself. And then you also have the apoptotic bodies, which are also showing up in the low frequencies. So what we can do is generate a metric. In this case, we call it an apoptotic index. And for instance, when you're in the shell, these apoptotic indexes for lots of these apoptosis-inducing drugs is positive, whereas in the core, they're actually negative. And so they're testing negative for this apoptotic index in the core because they don't have the energy to drive apoptosis. And then we've verified that with confocal microscopy. This is another example of just looking mechanistically. Mitosis is a fairly large event for us because I don't want to call it a little explosion because mitosis is actually pretty slow. But it's a large amplitude event, and it also has a specific Doppler signature. And so we can color code pixels where we see the signatures of mitosis. And you'll notice that it's all out here in the proliferating shell. Over here, we've applied Taxol, and so let's just actually run this one again. And so initially, we do get the mitotic events out in the outer shell, but because Taxol is shutting down mitosis, it actually shuts down those events that we see. And so we can track the decrease in the number of mitotic events for 2 different Taxol concentrations here. This is negative control. Mitosis is going down in general in the untreated spheroid, but not nearly as dramatically as with Taxol. I do want to point out that within a few hundred minutes, we get a full mitotic assay completed on this tumor spheroid. Whereas I just want to point out that tumor spheroids, even today, are used in a growth delay mode where you actually let the tumor grow for days, you apply a drug, it slows down the growth, you remove the drug, and then the growth speeds up again. This can take 3, 4, 5 days, and all you get is that it delayed growth, and you get no other information. We're getting the full spectral information, and in fact, we're able to pick out these mitotic events because of the spectral information, and we can even localize it to the fact that it's in the proliferating shell. All right, well, now I come to personalized cancer care. The pressing need is that cytotoxic chemotherapy is still the dominant standard of care. There's a lot happening with immunotherapy, and it is doing great things for many patients, but still the majority of patients are getting cytotoxic chemotherapy, and depending on the drug and the disease, only 30 to 60% of the patients receive no benefit from the chemotherapy. And so it means that you have cancer patients who are receiving chemotherapy, and it's not only not helping them, but it's making them sick. And it's because all patients suffer from the toxic side effects. Now, there is an interesting use case for a chemoresistance assay, and this is in a neoadjuvant setting. And so what neoadjuvant therapy does is that you apply chemotherapy to the cancer to reduce its size prior to surgery. And so if you have a much smaller tumor, or possibly the tumor disappears before surgery, then it means that the surgical incursion can be a lot more benign. But if you have a patient who is not responding to the chemo, not only is their cancer possibly continuing to grow while you wait for surgery, but you've also degraded the health of the patient before they even go into surgery. So if we could find that patients are going to be chemo resistant, then we could have them go directly to surgery. And so they're going to surgery faster, getting rid of the tumor quicker, and they're in good health when they go into surgery. And so it means that if we can identify patients who are chemo resistant, then they can bypass the chemotherapy and go straight to surgery. So the way that we run our clinical trials is actually fairly straightforward. We piggyback off the diagnostic biopsy, which is taken anyway by the doctor, and that's done for diagnostic purposes. And for that reason, we're actually considered to be non-invasive because we never go back in for another biopsy. And so just using some excess material from the diagnostic biopsy, we'll break that up into about 70 pieces, each about a cubic millimeter in size, put that into a multi-well plate, and then just apply a panel of FDA-approved drugs. And these would be the drugs that the doctor might be trying to select them on. And what we do is with our libraries and with some machine learning, we look for signatures of chemoresistance. And so I'll talk about these deep twin and triplet neural nets in just a moment. So let me dig into one of our clinical trials. We've done 4 clinical trials, but let's look at this esophageal cancer. It's a phase 2 clinical trial. It was done at the Indiana University School of Medicine, and our collaborating doctor there was Dr. Shadia Jalal. And so this is human esophageal cancer. We enrolled 28 patients, 16 were resistant, 12 were sensitive. The treatments were carboplatin plus Taxol in a combination, and then also cisplatin plus 5-FU in a combination. We also applied the monotherapies too, so carboplatin, cisplatin just alone. This is the layout of the 96-well plate. I do want to point out that when we break the spectrograms into features, if you take the number of patients, number of wells, number of features, we're dealing with almost 60,000 features in this clinical trial, and that's why we turned to machine learning. But before that, let's just look at the average over the resistant patients, and then just the average over the sensitive patients. So we're not predicting individual patients here, we're just averaging over the whole cohort, but what we get is a dramatic difference. And so these signatures between the resistant patients and the sensitive patients are striking. And you don't need neural nets to tell you that there is a difference in how the patients are responding on average to these drugs. But what we need to do is we need to do this on a patient-by-patient basis. And that's harder because there is a lot of patient-to-patient variability, and then you get into the heterogeneity. And so that's why we turn to neural nets. But before we do that, we have to generate feature vectors. And so most of the features are coming from these filter primitives, which are either global time frequency decomposition filters, or they're local time frequency decomposition filters, and they fill in these elements of this feature vector. And so we typically will work with a 40-element feature vector that have come from these spectrograms, but also things like the backscatter brightness, Nyquist floor, the knee frequency, things like that come from the spectrum. All right, so we like to work with triplet loss neural nets. The reason for that is that these are, well, let me say that it's driven by our application. So when we're looking patient to patient, the differences between a patient who's sensitive or resistant to the chemotherapy can be fairly subtle and on top of a high background. And so triplet loss neural nets are very good at common mode rejection. Common mode rejection is a phrase that comes from electrical op-amps. It's if you're amplifying a differential inputs into an op-amp, if there's common noise on the two inputs, the op-amps are really good at subtracting them away. Well, these triplet neural nets do roughly the same thing. And so it gets rid of the common parts of the signal, and it's just zeroing in on the differences between being sensitive and resistant. These are fairly simple neural nets too. So this isn't like the Google nets and the other deep networks that people are using. These are actually fairly shallow and fairly simple neural nets. Just feed forward neural nets, but this triplet loss actually makes them pretty powerful. What they do is they reduce dimensionality. And so I mentioned that we have a 40-dimensional feature vector, but what we want to do is reduce that down into a more manageable dimension. And 3 is good because it's easy to visualize. So what the triplet loss can do is it can lower the dimensionality into this 3-dimensional latent space. Now, this is just an example on MNIST handwriting data sets. These are famous for training neural nets, and we can just let this run. Initially, all those colors, there's 10 colors because there's 10 digits. They're all mushed together. Now they're separating. So this is as the neural net is learning on training sets. It's creating these point clouds that are separating. And so 1s don't look like 8s, and 7s might look like 2s. And so the 7s and the 2s will be closer together. 1s and 8s will be much farther apart. And as this runs, and we've only done about 400 epochs here, you'll notice this pretty nice separation. But this is the MNIST data set, but we want to do the same thing then actually with our cancer data. So this is where we did a 3-class classifier. So we're in a 3-dimensional latent space, but we also have 3 classes. And so sensitive, resistant, and also partial response. And so those are the 3 colors here. And we do get this nice separation in latent space between the reds, the blues, and the greens. The BDI predictions are given over here for individual patients. So there's 28 patients here. Now, there were 10 patients who did not get surgery. And while we do have other information about how they responded to their drug, for this clinical trial, we only looked at patients who went into surgery because their tissues went to a pathologist that were then graded. And so there's grade 0, 1, 2, and 3. We've lumped 2 and 3 together. These are the resistant patients. Grade 0, these are the patients who responded completely to their drug, and grade 1 was just a partial response. What you'll notice is that we got all of the sensitive patients, and so it means that we have 100% negative predictive value. So these patients were testing negative to chemo resistance. On this side, these are the patients who really need to bypass chemotherapy and go straight to surgery, and we did get 2 of these wrong. But this is not the final story. We're still working on the neural net. There is a lot we can do in terms of training, the depth of the net, a lot of hyperparameter tuning. So we're still working on seeing whether these patients had something intrinsic that made them look sensitive when they weren't, or whether it's some aspect in the neural nets and the way that we condition the data before training the neural nets. And so that's still work in progress. So we've completed 4 clinical trials: human ovarian with 20 patients, human breast with 55, human bladder with 30, and then the esophageal, which I've described, with 28. There's a lot of different ways we can visualize this. This is a network, a similarity network way of visualizing the patients. This is just looking at probability of being sensitive or resistant. This is a way of breaking down cohorts with a sort of a 4 class classifier, and so this is actually officially known as Onco4D, and then I talked about these. In all of these, we're running around 84 to 90% accuracy. That seems to be pretty consistent across all these clinical trials. So we also had a preclinical trial in canine B-cell lymphoma. So that was a 5th trial that we did. And there too, the accuracy was around 84%. So it does look like we do miss 1 patient in 10 or 1 or 2 patients in 10. But in a neoadjuvant setting, if we're wrong, what happens is the patient just goes straight to surgery. And so that means that the patients are getting treated for their cancer, and it may even be a better treatment to just have the surgery faster for those patients. And so that's why in this neoadjuvant setting, having an 85% accuracy is probably something that would help the clinicians get better outcomes for their patients. And I do like to point out that Cologuard, which is FDA approved, and you see it in commercials every night on television, Cologuard is only about 85% accurate, and it has a major market that they're marketing to. And so there is actually clinical value to having a test that may be only 85% accurate, but we are identifying patients who should be steered away from their chemotherapy and get better treatments. I want to finish this with a sort of looking forward. Currently, we have to build libraries for each new disease and each new type of tissue. It would be a lot faster and less expensive if we could come up with universals, if there was some kind of universal signatures. So as a first step towards that, we did a comparative oncology study between human esophageal cancer and canine B-cell lymphoma. And I want to point out that this is going cross-species. This is human and canine, and it's going to very different cancers. This is epithelial cancer, that's esophageal. And this is actually a blood cancer, although our biopsy is a solid biopsy because we do biopsy the lymph node, but still, this is actually a blood disease. When we did unsupervised clustering to generate the phenotypes, different phenotypes and how the tissue is responding to drugs, we did the same technique for the humans and the dogs, and we got almost identical results. And so the 4 dominant phenotypes for the humans, and you look at the 4 dominant phenotypes for the dogs, and they're virtually identical. And so that raises this possibility that there are universals. And we have a long way to go on this, but because we're not predicting patients over here, we're just, in this case, just finding general classes of behavior. But it's a first step to actually seeing if there are some universal signatures, so that in the future, we possibly would be able to train a neural net that would be able to predict patients with a new disease and new drugs that the neural net was not trained on. So digital holography in optical coherence tomography has a lot of use cases. I've just talked mostly today about the personalized cancer therapeutics, but it could also be a companion diagnostics for drug discovery. We are working on bacterial and viral infections, and we also have projects on artificial reproductive technology, or IVF, and where we can look at embryos and try to select the most active embryos for transfer in utero. And so in conclusion, I want to thank my team, and we have a growing number of doctors who are part of this research with physicists, so it's a great interdisciplinary collaboration. And then a complete aside, but because this is a talk on digital holography and interferometry, I do want to say that I've had a long-standing interest in the history of science, and so I read a lot about it, and I write a lot about it, and so I've published a definitive history of interferometry and holography. This is actually a non-technical history, so it reads almost like fiction. And some of the stories of some of the scientists and what they had to do to make their discoveries and get their discoveries accepted, it actually, there's some good stories in there. And so this came out from Oxford University Press in 2023. And thank you.